Good morning or good afternoon, whichever class I'm working with right now. Let's go ahead and zoom through the other 3 pages of this. Now, it's probably going to be, you probably won't be able to finish all of it today, but don't worry, you can finish it tomorrow. You do need to have the notes finished by Tuesday though, so please make sure you have that done. So, lipids, we've talked about carbohydrates, which are sugars. Lipids are just fats. Okay? And fats are composed of carbon and hydrogen. That's it, C and H. And they create these like fancy little chains that look like this. Okay, where carbon's in the middle, and then we have attachments of H or hydrogen all along the edges. Now we're going to find out later, the shape of these depends on where our hydrogens are actually placed. So, these are what I just drew is called a hydrocarbon chain. A hydrocarbon chain. It's, if we think about the shapes that our macromolecules can make, this is going to be a straight shape. Now we're also going to find that it does have it can be more like straight, or it can be like what we call kinked, where it has like a little kink in it, but we'll find out that later. So a couple functions of lipids. Long-term energy. Which, with carbohydrates, we had short-term energy. That's what our body burns first. Well, when we take out all of the carbs in our diets, our body switches over to the fats, where we have our energy stored. And it also creates barriers. Okay, protection. Fats are nonpolar. Okay, fats are nonpolar. So they're going to repel water. Or at least part of them's going to repel water anyway. Now, we've talked about monomers. Now, remember, a monomer is that one single part. It's like a brick of a house. It's that one basic part that can't be broken down anymore. Well, it can be, but it's that one basic part for that molecule. So, in lipids, the monomers are called triglycerides. And we also call them fatty acids. I'm sorry. The monomer is a fatty acid, not triglyceride. The monomer... There we go. Sorry about that. The monomer is a fatty acid. Now, triglycerides are basically a polymer. Okay, so it's a bunch of monomers put together. And the triglycerides are stored in fat cells of the body. So if you go to the doctor and they say your triglyceride levels are high, that's not a good thing. If we go to the store and we go to the cooking section, we see all different sorts of fats. We see solid fats and we see liquid fats, so fats or oils. We see solid fats or oils. We see liquid fats and oils. So if they are an oil, so if they're liquid at room temperature, then they're known as an oil. If a fat is liquid at room temperature, it's an oil. So like vegetable oil, or sunflower oil, or canola oil, or avocado oil, or truffle oil, or whatever types of oil you go after, corn oil. All of those are called oils because they're liquid at room temperature. But if they are solid at room temperature, okay, they're known as a fat or wax. So if you've ever seen vegetable shortening or manteca, it's hard. It's hard at room temperature. It's solid. Coconut oil, okay, it does come in a liquid form, but it also comes in a solid form. Still oil, but it's the reason it's solid or liquid at room temperature is due to the molecular makeup of those lipids. Now, there are two types of fats, saturated and unsaturated fats. So, saturated fats, well, before we go in there, let's talk about the structure of a lipid really quick. It has two parts. It has the head and it has the tail. The head is polar. Okay, so remember, we call it hydrophilic. Okay, hydro means water. Oh, don't want that. Hydro means water, and philic is love. So this is water, and this means love. So it loves water. The tail is nonpolar. Okay, so again, just the opposite of polar. It doesn't like water. So we actually call that hydrophobic. We call that hydrophobic. Hydro meaning water, phobic meaning scared of. So hydro, water, phobic, scared. Now, all of our fats have this sort of shape to them, where they have that polar head and that non-polar tail. And we have two types of fats that we work with. We work with saturated and unsaturated. Both are in the types of foods that we eat. Now, saturated fats are bad. Saturated fats are bad. The reason being is their molecular shape. If we look down here at the lower left, I have it highlighted in green here. The reason saturated fats are bad is because they create a straight chain. And because of that straight chain, they can get caught in our arteries a lot easier than, say, something that has some sort of shape. So these are the bad fats. Bad fats. Our good fats are our, I'm going to highlight in blue here, are our unsaturated fats. And the reason they're good or better is due to the fact that it has this bent shape to it. It like goes down and then it bends. And because it has this bend in this molecular shape, it can't get caught in our arteries as easily. So these are good. So let's talk about saturated fats specifically. Saturated fats are have tails with only single bonds. Okay, by single bonds, I mean that's a single bond. It's a bond between a carbon and a carbon. It's not a double bond. And I know we didn't go over double and single bonds, but it has to do with the number of electrons they're sharing. So again, carbon and carbon, and there's hydrogens all around that to make up and make everybody happy. So there's only single bonds between the carbons, which means there can be no hydrogen bonds. I'm not sure what I meant there. I'm going to have to look into that. Because hydrogens are going to be on the outside here. Unsaturated fats, okay? Tails with at least one double bond of carbon. And this creates a kink or a turn in the tail. Again, saturated fats are good, unsaturated fats are bad. So, if you take a second, pause the video, go to your Macromolecule graphic organizer, and on the front page, you can fill out the information from this page that you just did onto that graphic organizer to help visualize what's happening. All right, let's go ahead and move on into proteins. Proteins are fun. There's a lot of moving parts to proteins, but it's really not as scary. I'm going to go into a little bit more detail on these notes than I necessarily want you to know. And I'll point out the things that I really, really, really want you to know before or as we go through here. So, proteins are made of small carbon compounds that we call amino acids. Now, remember, amino acids are those monomers here. Okay? They're those single units. And this molecular picture that you see down here in the structure, that's the monomers. So, the elements that exist inside proteins, you can see there's quite a few here. Carbon, hydrogen, oxygen, nitrogen, and sulfur. No phosphorus. Hint, phosphorus is actually only found in some lipids as phospholipids and then in our nucleic acids. So the primary building blocks, proteins are the primary building blocks of our bodies and of living organisms. If you think about it, they make up our muscles, they make up our cells. As you're going to see here in a minute, their functions are numerous. They help support, so think about like our muscles, our structure. They help transport things. We talked a little bit about hemoglobin, how hemoglobin is the protein that's in our red blood cells that helps transport oxygen through the body. It can help speed up reactions, which that's going to be our next topic of discussion after macromolecules is enzymes. And then it's the basis of our hormones in our bodies. Now, structure of amino acids. Those amino acids, again, are monomers. Now, I do not want you to freak out about this next section. This is a molecular molecule, okay? It's like we call it the stick and ball model. And you're not going to have to memorize every single part. But what you are going to have to know is the general shape. So this molecule is shaped a heck of a lot different than, say, the ring from the carbohydrates or the straight line from the lipids. All right? And it's got several different pieces. And here I want you to get a couple colors out because we're going to color a little bit. So our first section is this central carbon here. Okay, this is our central carbon. Okay, and it's going to be green. It is the center. There are pieces of this that are going to be the same in every single protein or every single amino acid. And then there's one thing that's going to be different. It makes everything different. Okay, then we're going to highlight our hydrogen. The hydrogen is the second piece here. Okay, hydrogen. Okay, then we have what we call a carboxyl group. It's carbon and oxygen and a little bit of hydrogen. And I bet you can figure out where that's at. Carbon, oxygen, and hydrogen here, this side. Okay, it's called a carboxyl group. Again, this one's on every single amino acid as well. Our next is called the amine group. The amine. Amine is where the amino acid gets its name. So I'm going to color this pink. It's right here, amine. Okay, it's nitrogen and hydrogen. And that amine is always NH2. Okay, now we have that ARG. Now, R doesn't really stand for, it stands for radon. No, it doesn't, that's RD. Anyway, R is not really anything. R is what we would consider a variable. So let me find a good color. Let's use, ooh, all those are ugly. Let's use gray. I don't like gray, but. Okay, this R. R is what we call the variable. Okay, there are 20, oops. There are 20 different amino acids in our bodies that are available to be used. Now, the only thing that makes each of these 20 amino acids different is this R group. Everything else is the same. So, in methionine, which is our start, which starts making our proteins, it has a different R than, say, lysine does. So, later on when we talk about how proteins are created and how proteins are made, we're going to go into that. But know that it's structured like this. It has that variable R group in it, and there's 20 different amino acids. So, again, this variable, 20 different amino acids, and when multiple acids are bonded together, okay, we get this polypeptide chain. And you can see here we have that amine group, and our the mosquito, that amine group on the other side here, that NH right here, actually combines to create the bond between two amino acids, which we call a polypeptide chain. Again, this right here we'll worry about later. Okay, bonds between amino acids are called peptide. Okay, peptide bonds. They form between the amine group and the carboxyl group. So, your main ideas that you need to know here is the structure of this amino acid, the functions, its building blocks, so the monomers, and the elements that are in it. Later, we're going to talk about how they bond together and how they create such unique and different proteins in our bodies. All right, our last set of notes, nucleic acids. These are kind of fun. I love nucleic acids. I like DNA. I love the genetics part that we go through. So, let's run through these fairly quickly. So, nucleic acids. There are several different elements to them. I bet you can guess at least one of them's H, one of them is C, and one of them's O. CHO. Then we have nitrogen and we have phosphorus. Just like I told you guys earlier, phosphorus does exist in nucleic acids and sparingly in some lipids. Now, its function is to transmit genetic material. Now, this is specifically DNA. RNA actually is the blueprint to create proteins. So, you need RNA. RNA is actually made from DNA or read from DNA. But anyway, there's the two functions. Now, we're going to talk about the monomer or the nucleotide. Okay, the nucleotide. The nucleotide has 3 parts. And I'll say it time and time and time again. Sugar, phosphate, base. Sugar, phosphate, base. sugar phosphate base. So let's color. We're going to color the first, the square blue. Now, I don't really care what color you color it, but this is our phosphate base. You guessed it. I bet it has a phosphorus in there somewhere. And it does. That's where it gets it from. Okay, and then we have our sugar. Now, the sugar here, which we're going to learn later, is actually called deoxyribose. And it's the sugar found in DNA. And earlier when we were drawing carbohydrates, I was drawing carbohydrates with 6 sides, or sugars with 6 sides. And yes, that's accurate. Glucose is a 6-sided sugar. But this is what's called a pentose sugar. Okay, a pentose sugar, where it actually only has 5 sides. So this is the sugar. Okay, for funsies right now, I'm going to write deoxyribose, because that you'll have to know that later on when we talk about the difference between DNA and RNA. And one of those is that DNA has deoxyribose in it. Okay, and then we have our nitrogenous base. Nitrogenous bases are fun. There's a whole history that we kind of learn behind them that's really intriguing and kind of makes me mad. So, nitrogen base, and I bet you can guess it. What element is found in there? Nitrogen. So, there are actually, depending on if you're looking at DNA nucleic acid, or if you're looking at RNA nucleic acid, there are different bases. All of the bases are listed here. In DNA, there's A, T, C, and G. In RNA, that T goes away and gets A, U, C, G. So that's why I have all of them listed here. So A stands for adenine. You might not need to know these 100% now, but you will need to know them in our DNA unit. Guanine, thymine, cytosine, and uracil. So these are the variable parts. If we think about like our protein, this is the variable part for nucleic acids. And the way that, and this is a little bit going into a little bit too much, but the way that these DNA molecules actually bond together is through like a stepladder motion. So you have a base, you have a sugar, and then you have your nitrogen. And your next nucleotide is bonded like this. So really what's bonded together is your sugars and your bases. And when I say base, I'm talking about the nitrogen base, not the phosphorus base. So the nitrogen base. And the sugar and the phosphate actually create what's called the sugar phosphate backbone. So later on, we'll delve into the structure of DNA and why it's structured that way. Now, I've said over and over and over again that our body uses carbs and lipids to create energy. And our cells are using that glucose, C6H12O6, to break it down into several parts. And part of that is ATP, or adenosine triphosphate. Now, ATP is extremely, extremely important, and it is a very, very powerful energy for our cells. So, ATP has 3 different phosphates. Adenosine triphosphate. 3 phosphates attached together. Now, the fun thing about phosphate is they don't like being in groups of 3. They only like being in groups of 2. So our bodies will actually force 3 of them together to hold energy. And then when it needs it, it will break one of those bonds, as you see in this picture. It will break this bond between two. And when you break a bond, you're releasing energy. So when a bond breaks between a phosphate or between phosphates, energy is released. And that's the energy, when I talk about cellular energy, that's where we're getting it from, is that ATP being busted apart. Now, our bodies will take energy, force those phosphates back together to hold energy. It's kind of like a wallet, where you put your money in your wallet, and then when you break it out, you buy something. That's kind of like what our body does. Okay, our body stores money into these cells, okay, energy, and then when it needs it, it breaks it out. So, hopefully, you can finish this in one day. It's not a big deal if you don't. It's not homework. Please email me with any questions, and there will be a reading guide. It's not like the reading guides we're used to doing. It's a little bit different. So, stay tuned for that. That's going to be Tuesday's work, part of Tuesday's work. If you have any questions, please feel free to email me or message me on Google Classroom. I hope you guys have a wonderful day.